Good afternoon, students. In today's class, we will discuss about mutation. Now, mutation is something, a deviation in an individual from the normal wild type. Let's see at the image, the dog, the bull, the cat, even the humans, they have some deviation from the normal individuals present in our local community. Now, how this actually mutation occurs and what are the reasons behind which make the mutation to prevail in the human society, we will discuss in due course of time. Now, Hugo de Varis was the scientist who first coined the term mutation. Who have an X and Y chromosome. When this X and Y chromosome is divided by the number of autosomes. That means the individual has half the number of X chromosome as compared to the previous case. Over here the ratio is 0.5, but since this individual carries a Y chromosome, this will be a fertile male. In another scenario, if the individual carries only a single X chromosome and no Y chromosome is present, then also if we divide a single X chromosome by two autosomes, the ratio will come to 0.5. But this individual will be a sterile male. Now let me tell you something about this Drosophila melanogaster community. Now what actually happens, there are mostly sterile male present who act as a worker. There is a fertile male present who will help in the reproduction of the fertile female. Coming to another case, if there are two X chromosomes and a single Y chromosome is present and two autosomes are present, then also the number of X chromosome by the total number of autosomes will give you a ratio of 1 and hence it will be a female. If in certain scenario two X chromosomes are present and three autosomes are present, then that ratio will be above 0.5 and below 1. Such a scenario will lead to the development of intersex species. Now look at the image towards the left hand side of the slide and at the bottom section. What is it written? That ratio of X is to A, if it is greater than 1, then it is a meta female. That means the female will have developmental problems. If the ratio of X is to A is less than 0.5, then it is a meta male. That means it is a sterile male. And if the ratio is between 1 and 0.5, then it is an intersex. That means it will be a mixed progeny. Coming to the image towards the right hand side of the slide, the bottom image, it gives you a accounting of the number of X chromosome compared to the number of autosomes, the expected ratio and the phenotype of the individual. Here you will get a glimpse of what different kinds of ratio can exist in this Drosophila melanogaster community and what will be their individual phenotypes. In our previous video, we have discussed up till linkage. The sums of the linkage I will upload in a couple of days. Today's video, we will discuss mainly about genic balance, mutation and ploidy states. Coming to genic balance, before we discuss about this topic, we need to first understand that in humans and certain insects, the male and female distinction is done based on the presence of sex chromosomes. We all know that in humans, if an individual carries 22 pairs of autosome along with double X chromosome on the 23rd pair of chromosome, then that individual is a female. Similarly, if the 23rd pair of chromosome contains XY chromosome along with 22 autosomes, then that individual is a male. In Drosophila, which is a model organism for studying the morphology of humans, they have a very different calculation based on which the sex of Drosophila is determined. If you look at the image towards the right hand side of the slide, X by A indicates the number of X chromosome divided by the number of autosomes. If this ratio zeroes down to 1, then it is a female. And if this ratios comes down to 0.5, then it is a male. Now, if we define genic balance, it says that in Drosophila melanogaster, the sex is determined by the ratio of X chromosomes and the autosomes. Coming back to the image towards the left hand side, see in the first section they have said that if double X chromosomes are present along with the autosome, and if the ratio is 1, then it is a female. If X and Y chromosome is present along with autosome, then obviously half the number of chromosomes, sex chromosomes are present in this individual compared to the previous one. Over here the ratio will be 0.5. That means this individual will be a male. If this individual, let's discuss about mutation. What mutation is? A change in the nucleotide sequence of a particular genome. The image on the right hand side of the slide shows the DNA structure where the nucleotide sequences are altered. If you remember, DNA has 4 nucleotides: adenine indicated as A, guanine indicated as G, cytosine as C, and thymine as T. Now A always bonds with T and G always bonds with C. This mechanism is regulated by an enzyme present inside our body. We will discuss the details of all these mechanisms in the next chapter by the name molecular inheritance. For the time being, let's understand that A bonds with T and G bonds with C. If there is an alteration in this pattern, it will lead to genetic mutation. Now, with every round of replication, there is a chance of mutation. But our cell has a repair system which prevents the mutational changes. For example, if the G suddenly start bonding with T or the A suddenly start bonding with C, this will lead to a mutant phenotype. So, we can describe mutation as a sequential change in the nucleotide sequence which results in the failure of the system to revert the change. Now, when this failure occurs, with every round of replication of DNA, mutational changes occur. Our system has a repair mechanism which heals the mutation, but those repair mechanism falls down at 1 in 10 million times. Now, once they fall down, the mutations start accumulating inside our body. Now, mutation are usually of 3 types. It can be a silent mutation. It will not show any change in the body. It can be a lethal mutation. It will show some disease phenotype inside our body. Or it can be an advantageous mutation where it will add an advantage to your body compared to other individuals. Look at the image towards the left hand side. There is a bacteria which is harmful for the humans. So what we do? We consume antibiotics to kill the bacteria. Now, if we do not take the prescribed dosage of antibiotics, suppose a doctor told you to have a 7-day course and you suddenly stop taking the antibiotics at day 4 because you started feeling healthy, then the remaining bacteria will convert into a mutant phenotype, and this will lead to further side effects of the disease which won't be cured if you take that antibiotic again, because these bacteria are mutant. They won't have any effect of the upcoming antibiotics. This leads to a scenario in our community known as antibiotic resistance, because of which penicillin cannot be taken by most individuals because they have developed a resistance to penicillin antibiotic due to misuse of the antibiotic. Further, if you look at the images towards the lower part of the slide, the Hulk from Avengers and the Wonder Woman from the X-Men series, they are although fictional characters, but they have been formed due to certain mutation in their genetic makeup. Mutation in an individual can take place at any point of time in their life cycle. Now, mutation are usually of two types. One is known as somatic mutation, and the other one is known as germline mutation. When somatic mutation occur in an individual, it can occur either in their fetal condition or in their due course of time while the individual develops. Those changes or those mutational phenotypes will be revealed only in that particular individual in due course of time. But if the mutation takes place in the germ cells, then it will affect two kinds of cell. In male, it will affect the sperm cell, and in female, it will affect the ova cell. Those mutations will not show the phenotype in the carrier or the individual who accumulate the mutation, but those changes or the germline mutation will be revealed in the future progeny. If you look at the image where the germline mutations are highlighted, see the sperm cell have got mutation, and once it mates with the egg cell, it leads to the development of an individual who has mutational characteristics similar to the sperm cell. But if a somatic mutation takes place, then it will be revealed in the individual only at a particular part of the individual's body, but not in the future progeny or the parental generation. This slide describes the two types of mutation: transition and transversion. Now, in our DNA, the bonding always occurs between a purine nitrogenous base with a pyrimidine nitrogenous base. We all know that adenine and guanine are purine nitrogenous base, and thymine and cytosine are pyrimidine nitrogenous base. Now we know that adenine will always bond with thymine, that means the purine will always bond with pyrimidine, and guanine will always bond with cytosine. Similarly, another purine will bond with another pyrimidine. Now, in a transition type of mutation, the purine will always change into another purine base, and the pyrimidine will change into another pyrimidine base. See the image B in the right hand side of the slide. The AT bonding, that means the purine pyrimidine bonding, changes into GC bonding, that means again the purine pyrimidine type of bonding. Although the nitrogenous bases are different, but the bonding pattern is same. The purine adenine has mutated into purine guanine, and the pyrimidine thymine have similarly mutated into purine pyrimidine cytosine. In a transversion kind of mutation, the purine will change into pyrimidine. See the image. The purine adenine has mutated into pyrimidine thymine, and the pyrimidine thymine has mutated into purine adenine. This kind of mutation is known as transversion. This particular slide describes the various kinds of mutation. Now the image labeled as A indicates point mutation. Now what actually happens in a point mutation is the nitrogenous bases changes at a particular nucleotide sequence. For example, UUA, which is present in the original DNA. Now let me tell you, U is present in the RNA and not in the DNA. U stands for uracil, and in the RNA, in place of thymine, uracil is present. Now if UUA is present and another nitrogenous base gets incorporated between uracil and adenine, see in the image where it is mentioned frame shift mutation, that means the entire frame is altered. Now how actually this frame shift mutation takes place will be discussed in the later chapter. Coming to silent mutation, where the initial U is altered into C, that means CUA. Now silent mutation, as I have told you, is a kind of mutation which do not show any particular phenotype in the individual. Now coming to another mutation which is known as missense mutation. Over here the U is changed into G. Now this kind of mutation leads to the change in the amino acid sequence which was originally not present in the parental DNA and the parental protein, and this amino acid will cause a lethal change in the individual. Hence, it is known as missense mutation. And then there is a nonsense mutation. A nonsense mutation means the protein synthesis mechanism will simply stop. So, see, UAA codes for a stop codon, which leads to the blockage of the mRNA and prevents the further coding of the proteins. Now the detail of how nitrogenous bases creates a codon and how a codon changes into a protein synthesizing machinery will be detailed in the next chapter. So for the time being, just try to understand that point mutation occurs when a single nitrogenous base gets altered or gets mutated to another nitrogenous base, and that can cause four kinds of mutation: frame shift, silent, missense, nonsense, among which silent mutation will not cause any alteration in the phenotype. Frame shift and missense may cause the synthesis of a new protein which may be lethal or it may be useful, or it may not cause any significant change, but the nonsense mutation will lead to breakage of the mRNA coding sequence. Now coming to the image towards the right hand side of the slide, there is another examples of nonsense mutation. Now this kind of nonsense mutation can lead to disease scenario. See image D towards the bottom of the slide, which leads to Parkinson associated gene mutation, and it causes Parkinson disease, which is related to a neurological disease, usually occurs in age old individual. Now let me tell you some facts about mutation. Now mutation do not really occurs in embryonic condition or in child. In our present environmental situation, certain diseases are known as cumulative disorder. For example, cancer. Now, you will rarely find any individual who is within 10 years or 20 years of age and they are suffering from cancer. Now, why this happens? Because cancer takes a sufficient amount of mutational changes to occur in an individual. Now, the percentage of people who are above 50, they have a higher probability of getting infected from cancer, or rather suffering from cancer, because in their 50 years of lifetime they have accumulated huge amount of mutation, and at this particular point of time the repair system of the body is unable to cure those mutations. But in an individual who is 10 years or 20 years of age, their repair system is so precisely developed that they take care of each and every mutation occurring inside the body. Slowly and slowly, as we age down, our repair system halts and it loses its effectivity. Hence, Parkinson disorder or cancer or Alzheimer disorder, these are usually found in individuals who are above 50 years of age. The image C towards the right hand side bottom of the slide, it represents certain different kind of mutation which usually occurs directly in a chromosome. For example, the first one is known as a deletion mutation. Now, if a particular segment of the chromosome deletes out, it is known as a kind of chromosomal aberration, then it will also lead to the change in the protein sequences since certain nucleotide bases are missing. Sometimes it occurs that a particular segment of DNA duplicates itself. For example, the ABCD has duplicated into another set of CBD. Now, sometimes it occurs that an inversion occurs in the sequences. Look at the image. The ABCD changes into ADCB, and sometimes reciprocal or translocation takes place. Now, these are various changes which are known as chromosomal aberration, and chromosomal aberration usually causes a change in the sequence of the nitrogenous bases arranged in the original DNA, and due to this change certain kind of shifting, for example the frame shift mutation, can also take place in this kind of scenario, and it will lead to the formation of another kind of mRNA which is not present in the wild type, and it will lead to formation of new protein, which is usually a mutant phenotype. In this particular slide, we will discuss about the change in the number of chromosome, which is known as ploidy. In the previous slide, we have discussed about chromosomal aberration, which causes a change in the content of DNA of a particular chromosome. Now, what happens in a particular individual? In a healthy condition, as we can see the image towards the right of the slide, both male and female have 46 number of chromosome. But in states where there occurs a change in the number of chromosome in a particular cell, that situation is known as ploidy. Now, ploidy condition is mainly of two types, euploidy and aneuploidy. Now, if we describe euploidy, it includes the addition or loss of a complete one set or more than one set of a chromosome in a diploid organism. And if we describe aneuploidy, there is also an addition or loss of one or more chromosome to a haploid genome. So, for euploidy, we are talking about a diploid organism, that means the entire organism. And when we are talking about aneuploidy, we are talking about the haploid genome. Okay. Now, euploidy is mainly of three types: monoploidy, diploidy, or polyploidy. Now, in monoploidy, it involves the loss of a particular set of chromosome, and in diploidy, it involves the same number of chromosome which is present in a particular individual, that is, we are diploid organism. Now, there is an interesting condition. If in some condition there occurs a monoploidy, that means suppose a cell, once it enters the S phase after the G1 phase, it becomes 4N. Now, the 4N cell divides into 2N and 2N, two particular cells, which is the mechanism for mitotic division. Now, if due to non-disjunction there occurs the segregation of the chromosome in such a manner that when the 4N segregates, it segregates into N and 3N. This kind of condition leads to a single cell which has N number of chromosome in place of 2N, and hence this cell will be known as monoploidy state, and another cell which has 3N number of chromosome, that will be known as triploidy state. These triploidy state of chromosome occurs in a scenario which is mentioned in the diagram towards the left hand side of the slide as polyploidy. Now, polyploidy is usually of two types, autopolyploidy and allopolyploidy. Now, autopolyploidy is described as multiplication of a chromosome of a single diploid nucleus so that the homologous chromosomes comes from the same species. Hence, autopolyploidy can be mentioned as presence of 3 chromosomes or presence of 4 chromosomes. That means a deviation from the normal diploid state, but it should come from the same species. Whereas allopolyploidy describe a hybrid condition between two different species. That means these particular individuals will also deviate from the normal diploid scenario, but the deviation will take place when the chromosome arises from two different set of species. For example, the wheat, the maize, or the bajra, the cereals which we take, they are all alloploidy state of a particular plant species. Now, to have a exact idea what autopolyploidy or polypolyploidy is, I will mention some of the links for the particular description where all these things are described in detail. All these things will be mentioned in the description box. Coming to aneuploidy, as we have mentioned that it is the loss or the addition of one or more chromosome to a haploid genome. Now, anapolyploidy is hypopolyploidy or hyperpolyploidy. The similar to the addition or deletion of a particular set of haploid genome. Now, in a condition where there occurs a loss of a chromosomal set, it leads to monosomy. If some cell have lost a particular set of chromosome, there will be another cell who will gain the particular set of chromosome. It will lead to trisomy. That means 2n minus 1 in one cell and 2n plus 1 in another cell. Both the situation occur simultaneously. In the same way, if there occurs nullisomy, that means loss of all the set of chromosome in one cell, then there will be another daughter cell which will gain both the set of chromosome. It will lead to tetrasomy. So, monosomy will be accompanied by trisomy. That means the parent cell, if there occurs non-disjunction, then it will lead to the formation of two daughter cell. One will be monosomy and the other will be trisomy. In another scenario, if the parent cell give rise to two daughter cell and one daughter cell has a nullisomy condition, the other one will have a tetrasomy condition. So students, over here we have completed mutation, polyploidy state and some genic balance theory. Now to have a detailed idea about all these topics, you need to go through Veerbala Rastogi as well as Willy publication Fundamentals Biology. Along with that, if you need any specific study material, then you can call me on the particular WhatsApp number which I have mentioned. Then over there I will supply you all the study material related with this topic. Hope you have enjoyed the entire video and that has cleared your basic knowledge on the particular subtopics discussed. Further, in the next session we will discuss about few sums related with linkage, as well as we will go through some of the genetic disorders which is present in a human cell. For the time being, take care, stay safe. Goodbye.